So in certain marine environments, invertebrate animals can't rely on photosynthesis, but they need an alternative energy source. So some bivalves can live in symbiosis with bacteria that use chemical energy for carbon fixation instead of sunlight, so instead of photosynthesis. So here you can see an example of one of these bivalves. This is a leucinid clam, and you can see these bacteria are present in these specialized cells within the gills of this animal. And we also find species of this same family in the fossil record. So this is a nympholeucina clam from the Cretaceous. So this chemosymbiotic nutrition makes it possible for dense animal communities to be present in areas of the deep sea where life would normally be quite rare. So this is an example of that. So the specific ecosystems I'm looking at are called cold seeps, and this is an example of that, actually. And at these cold seeps, they are areas on the sea floor where chemical fluids come up from the sediment at an ambient temperature, as opposed to the hydrothermal vents, which are very hot. So we find these cold seeps in the fossil record, as we've also seen yesterday. So these are some more examples of such leucinid clams, and cold seep in Japan. So these species have modern chemosymbiotic relatives, but at pre-Cretaceous seeps, we also find bivalve taxa that are now extinct, as well as very dense brachiopod assemblages, singular species brachiopod assemblages like this one. So modern brachiopods aren't chemosymbiotic or associated with cold seeps, but their high abundance could be an indication of a chemosymbiotic lifestyle. So a bit more on chemosymbiosis. There are several types of chemosymbiosis. So this is a kind of a schematic of a cold seep. And there's the methane kind of fueling everything, coming from deep within the sediment. So the first type of chemosymbiosis is methanotrophy, which are bacteria that oxidize methane, and this electron transfer releases energy. So that is one type. And then some of this methane is anaerobically oxidized in the sediment by bacteria and archaea. And this releases sulfite. And the second type of chemosymbiosis is thiotrophy. So these bacteria oxidize the sulfite to produce energy. So most of the kind of the seep clams, which are multiple families, they're all thiotrophic, so using the sulfites. And then there's the one family of the seep mussels that can be methanotrophic or thiotrophic, or use both types of bacteria together, which is known as a dual symbiosis. So there are several ways to identify chemosymbiosis in living animals, but there's currently no direct evidence for chemosymbiosis in the fossil record. And we're investigating this by using stable isotopes. These are very useful because the stable isotopic values of these sources are very low, so for sulfur and for carbon of the methane, compared to the sources that the filter feeding bivalves use. So because of these very depleted sources, so we expect the tissues of these animals to reflect these sources, because they show the food sources that these animals are eating. Obviously, in fossils we don't find that many soft tissues, well none, but instead we can look at the shell-bound organic matter. So there's an organic framework that is present within the shell. So it's a framework around and within the calcium carbonate crystals of the shell, which you can isolate and then analyze. So we're trying to use this as a proxy for nutritional strategies. Because this shell-bound organic matter is deposited by the soft tissues, it is expected to also reflect these depleted sources, and which will then indicate chemosymbiosis. So most of this talk is going to be on, not on methodology, on stable isotope results, but other parts of the project are involved with a method comparison of how to isolate these organics from the shell, as well as some more questions about preservation, which I'm happy to talk about afterwards. Because no framework really exists for using this proxy, there's some basic research questions that need answering about if these different nutritional strategies are reflected in the soft tissues of the bivalves for carbon, sulfur, nitrogen, and how this relates to the shell organic values, and if we see a similar isotopic signal in the fossil record. So I'm first going to show some carbon and then move on to sulfur and nitrogen. So this is maybe a familiar plot. So these are some soft tissue values for different nutritional strategies. And you can see there's some clear differences from heterotrophy to pleiotropy, and then the very depleted values caused by using methane by the methanotrophs and the Gill symbiosis. So these are soft tissues, and then we can plot the shell organic values on top. So in this graph, so these are the karma values, and then in this graph, every column is a different species, and every point is a different specimen that I analyzed. So you can see these correlate very well, and you can see the distinct differences between the species. So then we can, what we've all been waiting for, the fossil values. We can kind of use these ranges kind of as a guideline to look at some fossil values that are analyzed. So below here are the ages, and then each column is a locality with some different species with suspected nutritional strategies based on their modern relatives. And it kind of it agrees with the pattern that we see in the modern species. So we have these kind of green circles, which are the suspected pleiotrophs. They have quite depleted values and also fall within the modern pleiotrophic range. And then we see some of these heterotrophs, so the yellow diamond things. They have much less depleted values. And then we see these chemosymbiotic mussels that have much more depleted values, even. And then some sea brachiopods that are analyzed that are showing lighter values, which might indicate heterotrophic, so filter feeding lifestyle. So in addition to the total shell organic matter, we can focus on a specific pool, which is the intercrystalline shell organics. So they are within the crystals, so they should be better protected and might be better preserved. So this is only a few values of that, but we can see a distinction here between the suspected pleiotrophs and then the one lonely heterotrophic value, but there's more to come. And hopefully this will also give us this distinction between the nutritional strategies. And for the intercrystalline, not bomb, S-bomb, we can check whether these organics are indeed original by looking at the amino acids, so the building blocks of the proteins that make up most of the shell-bound organic matter. So these are so for these amino acids, you can study them in a few different ways, and this is a way of looking at the different compositions. So in this graph, each point is a different amino acid. So if the material is original, we expect similar amino acid composition through times for these different bivalves, which is indeed what we see for these three examples that I pulled out, because it agreed for most of the samples. And we can especially see that when we have a contaminated sample here, where you can see that the highs and the lows are very different. Especially here the serine peak, which is something that we find in modern samples, so it clearly points to some contamination, but it confirms that the other samples are original and that the protein is original. We can also look in more detail at valine. We can look at a process that's called racemization. So in living animals, all amino acids are in a certain configuration, the L configuration, and after death, these slowly turn into a D configuration, which is the mirror image of that amino acid. And this happens at kind of a known rate, so the DL ratio is used as a geochronological tool. And we can see that our samples, that the order of the DL ratio increases with the age of our samples, which confirms their originality. So we have these subfossil samples all the way down here, with a very low ratio, then the Pleistocene, Pliocene, and then the older sample up there that are in equilibrium. So this confirms that the material we are analyzing is original, which is quite exciting since it's also a Cretaceous sample. And briefly on to some sulfur nitrogen. So we see this clear distinction in the soft tissues, which is mainly caused by the very low sulfide values that are around here, which we find for the pleiotrophic specimens. So again, we can plot the shell organic values on top. And we can see that they agree very well for nitrogen, but they're a bit less extreme for sulfur. But we can still see the distinction quite clearly. So this is just supporting 3 points of fossil samples for now, but more to come. And these are suspected thiotrophs, and they fall within the range kind of that we expect. So moving on to some conclusions. We can see that these soft tissues can be used to differentiate these three kind of nutritional strategies for carbon, sulfur, nitrogen, and that they mostly agree for the shell organic values as well, and that these can be used as a proxy in modern samples to differentiate these nutritional strategies, in particular chemosymbiosis, and that the values that we saw for the fossil samples agree with these observations for the modern ones, but we need to do some more preservation work to confirm this. But the initial results are very positive, and if we can use this proxy, it will significantly increase our understanding of these fossil gold seeps as well as other ancient marine ecosystems. So thanks for everyone who gave me shells, beautiful shells that I all crushed up, and thanks for the funding. So thank you for listening.